So yeah, today I'm going to talk to you guys a little bit about what Wheeler Bio is doing and how we're integrating machine learning into CMC development and clinical manufacturing for antibody and antibody-related modalities. So just a little bit of background on Wheeler. So we're a high science, high touch CDMO. We're located in Oklahoma City, Oklahoma, and we've got services that kind of span the biologic spectrum. One of the things that we focused on, so Wheeler is relatively new, was really identifying and outlining the CMC milestones that an innovative drug developer needs to get from discovery to sort of first in human clinical studies. We've termed it modular CMC, and each of the work packages essentially serve as a basis for scopes of work. So we need CMC milestone X, Y, and Z, and they're broken up into discrete packages. We also started off and we had a clean slate and we said, okay, what does the market need? And it's, in the antibody and antibody-related modality space, it's all about speed. So how fast can you go from a concept, target identification, lead identification, to having material to put into humans for the first time? Quality is non-negotiable, right? It's got to have the right product quality attributes. But quantity is important. So what we really strove to do was right size our manufacturing approach for early stage clients. Obviously the process has got to be scalable, and really our goal was to say, okay, with certainty, here's how long something is going to take and here's how much it's going to cost. Today I'm going to talk a little bit about how sort of AI and ML are enabling sort of efficiencies, shortening of timelines, lowering of costs. And so just a brief background. So AI, we're creating smart machines that mimic human behavior. So right now, Wheeler, we're not doing that. We're focused on the machine learning approaches, and this is essentially algorithms that enable computers to learn data without explicit programming. Underneath that is sort of deep learning, and one of the things that sort of encompasses all three of these is data science. So Wheeler invested in a small data science group, and we believe that this actually allows both time and cost savings during the development pathway. So what we want to do is use the data that we have and we've generated for programs that we've never seen before. So we don't want to reinvent the wheel. One of the very first things we did at Wheeler was actually evaluate cell line expression technologies and different cell lines, looking at a number of antibody and antibody modalities. And we were really dialing in a platform process. So we want to essentially obtain an electronic sequence and tell you, okay, within X number of weeks, we expect to have Y grams of your product. And so we ran about 1,100 bioreactor runs. Some of them were sort of screening technologies and then actually adapting and working out all of the workflows to go from pool generation through clinical manufacturing. And shown on the graph below is it's a graph of replicate error. And the easiest way to read it is the closer to zero it is, the more stable of a process parameter it is. And so on the x-axis, you've got essentially everything we measure in a bioreactor. So viable cell density, viability, nutrients, metabolites. And what we were looking for was something that was predictable, something that was repeatable time and again, and particularly with regard to titer. And so we built a platform process based off of all of these parameters. And we're using this data now to further enhance and train additional models. We have a recent publication. I think it came out this month in Communications Engineering. It's a Nature portfolio journal, and what we did was use this data to take a look at two things. So enhancing selection criteria for high-producing clones, and then intelligent forecasting. So can we use data that we have in hand to forecast what a different bioreactor scale should look like? And we have the data to then say, the model has never seen this. How does this actually compare to actual? So here's an example of some of the early titer prediction for clone selection. So what's shown here, we've got 4 graphs. You've got a stable CHO pool that was generated. Then you've got 3 clones that were derived from that pool. And what we were doing here was saying, okay, we have the actual data. If we train the model, will it tell us, based on an early time point, what the titer is going to be at the end at harvest? And really what we're trying to do is use it as a way to down select what I would call, poor clones faster and get them off of the pathway. And for each of these examples, you can see, with some low level of error, we're able to actually predict that quite accurately. The other thing that we're doing with clone selection in particular is actually clustering. So if you're seeding single cell, 96 well plates, you typically have 5 or 10 of them per molecule, all with single cell clones. How do we identify, honestly, clones that are bad that we don't want to carry through? We don't want to waste time and money on reagents. And so we've also started clustering them in a way that allows us to know, we know which ones have moved forward. We know which ones clients have selected to move to a master cell bank. And so defining that population and describing the characteristics of it as early as possible. So those are the only ones that move forward in the process. We've also used these data sets for intelligent forecasting. And this is really interesting. So we have data sets that run from Amber 250, 3 and 10 liter bioreactors, 50 liter bioreactors, and 500 liter bioreactors. So internally, as Wheeler was building out its clinical manufacturing, what we did was we actually supported internally, 50 and 500 liter manufacturing run. So taking the platform process and actually demonstrating it end to end. And so on the top, you have root mean square error. That's basically how well a model is predicted to the actual data. In the middle, you've got sort of graphs of actual versus predicted. So and what we're doing here is asking the model to predict what the clinical batch, the 500 liter batch, is going to look like without ever seeing that data. So we have it, but the model hasn't seen it. And so if we just use the Amber 250 training data set, we've got very low root mean square error. It means the model is predicting really well. So it's a really high quality data set. If we add in, say, 3 and 10 liter bioreactors, what we see is that the root mean square error increases a little bit toward the end, but again, it's a pretty predictable data set to use. If we add in just one single 50 liter run, we see that root mean square error actually go up. And what it tells us is that for this model to predict even more effectively, it needs a little bit more of that larger scale data. But again, right, we're trying to use this to say, this model has never seen data from a 500 liter run, from a GMP clinical run, but can we use the data that we have in hand from development runs to actually be able to predict what the output is going to be? We've done this also for a number of other sort of characteristics for bioreactors and cell culture. So what's shown here is viability. So we're trying to predict the viability at the end of cell culture. You'll immediately notice that the root mean square error, the y-axis is different. So this is a harder variable to predict. There's more variability in this data set than, say, for titer. And so, what we see, particularly toward the end of production runs, we see the root mean square error bump up. So those predictions are less accurate. It actually gets a little bit worse if we add in the 3 and 10 liter training sets. So within those data sets, we actually had more variability. But interestingly, right, and as an example, we just add in one single 50 liter bioreactor run and we see the root mean square error drop. So again, the evaluation of the model compared to actual is telling us what data we actually need to generate to make these better and more efficient. And so, shown in the middle below, you've got what the actual data was from a 500 liter run that the model has never seen versus what was predicted using data sets right out of the PD lab. And because we're talking about engineered antibodies, this is not meant to be exhaustive in any way, shape, or form, right? All of these are modular. I've seen people talk here about formats that I had never even contemplated before in my mind. But as we engineer antibodies or antibody-like molecules for specific activities, the complexity increases significantly. We go from something that's 2-chain, perhaps 3-chain, to things that are much, much, much more complex. Some of these can have common light chains, some of them can have Fc fusion partners, and that could be, some type of cytokine, it could be an scFv. So there's all of these modular building blocks in the engineered antibody space, and it increases the complexity of being able to predict how these are going to behave, right? Because the one thing that we really wanted to be able to do, particularly with monoclonal antibodies, was say, we know a set time, a set sequence, a set cost for folks to get this from discovery, get your lead candidate, development candidate nominated, get the CMC development done, get your clinical material and into humans. And so, we've started generating data around these. And so as you can see, so it's a similar slide to the one we had on MABs, but the number of input variables increases. So, we're talking about, okay, the number of chains that are being expressed, the size of chains that are being expressed, what the desired stoichiometry is, going from something like 1-to-1 to something that can be more complex than that. And what we need to do is actually generate data in terms of taking a look at balancing chain ratios. So, the first thing that we want to do when we receive a complex, engineered antibody sequence and we're starting cell line development is really dial in those chain ratios. We don't want product-related impurities. We want those minimized as much as humanly possible. They're typically more difficult to remove through the downstream process, and ultimately that leads to lower yields for a particular target product. So getting it right early and being able to dial those in is really important. And so, what I'm showing here is some data on the right, just two molecule types, a 2+2 symmetric bispecific. These are both common light chain, or a 2+1 common light chain asymmetric bispecific. And it was a simple question, okay? If we have additional light chains that are needed, if we increase the gene dosage and the expression of light chains, do we actually see a bump in titer, right? Is light chain limiting? Because for us, we don't care for making extra light chain because of the purification process. What we don't want to do is have that limiting, and then you have, heterodimers lacking light chain or homodimers lacking light chain. And so we set up just a couple simple ratios in terms of transfection. So with the symmetric molecule, so knob chain, hold chain, light chain, so a 1-to-1-to-2 ratio for the symmetric. We also increased by adding in an additional copy of the light chain expression vector, so 1-to-1-to-3. And it was, somewhat surprising, but science is humbling for me. I'd assumed we give it more light chain, we're gonna see increases in titer. And that was actually not the case. So it's telling us there's more than sufficient light chain that's being made in these cells, and that if we add in more genes, it's actually deleterious. So we don't see titers as high. So whether it's replication or transcription, the cell is putting its energy into making light chain that's not doing anything for assembly. We did the same thing or something very similar for the asymmetric bispecific. And in this case, we had some data early on from transient expression experiments that indicated that whole dimers were being observed at a higher frequency. And so the way that we set this up, so a 1-to-1-to-2, 2-to-1-to-2, or then that same set of knob and whole chain plus additional light chain. And we saw the exact same thing. So these are the data sets that we're using that we will feed into these models as well. One of the things that we typically do off of this expression ratio screening is actually purify material over protein A, identify what and ideally how much of product-related impurities are there as we move forward, right? So this is essentially the starting point of cell line development. This is a stable pool expressing product, and eventually this will move into, single cell cloning, master cell banking, kind of so on and so forth. So, as the modalities become more and more complex, it requires us to actually generate additional data to understand how we do this chain balancing as efficiently as possible. And as I said, right, like this is the starting point for everything for your CMC, clinical, commercial development is the cell line. So you want to get this part right. You want to have the right chain ratios dialed in. So with that, I'll leave you guys with a couple conclusions and probably a couple minutes for questions. But just wanted to let you know, right? So Wheeler, we very much live into a very high science, high touch CDMO. I mean, we have some great technical experts, and we do this in collaboration with partners. That's something that's really important. We're expanding the use of AI and ML, and really, I joke about this with my boss, but it's probably true that I'm hired to put myself out of a job. Like I was a PD person, right? That's what we did. But the whole goal here is to eliminate as much process development and wet lab work as possible, right? We want to take the data, all the information that we already have, and use that to streamline this entire process. If that data is just sitting there not being used, it's a complete waste. We initially started working with partners and outsourcing some of the AI ML work, and this is something that we're actually bringing more and more in-house. In an ideal world, right, we'd get to a place where perhaps there's a digital twin and we're actually using this to make decisions on the GMP manufacturing floor. And in order to do that, right, we can't have really a black box to say, okay, well, the data goes in, this comes out, we made this decision. So we have to be able to describe that very well to regulatory agencies. And so that's part of the reason that, we sort of ran some pilot programs. They were very successful, and we're actually investing more into that at Wheeler. We've got 3 sites in Oklahoma City. We've got process analytical development labs. We have clinical manufacturing. That's the building in the middle. And then we actually have acquired some shell space for expansion. It's about a mile away, the tower at Convergence. So, we're looking to sort of grow in scale. And then I just wanted to say thank you. I know it's an early morning. I appreciate you guys coming. I hope breakfast is good. I hope the coffee has perked you up. I hope I'm not putting you back to sleep. And then, thank the team at Wheeler Bio that supported this work. So Deepika Verma, she's our Associate Director of Data Science and Digitalization. She spearheaded all of the work in collaboration with the University of Oklahoma. We've got Yuk, Krista, Mike, Alicia, and Sierra. They're the ones that ran all of those 1,136 bioreactors early in the days to develop the data and to develop the platform process. And then the folks at the University of Oklahoma really, we were utilizing them for their sort of computer science skill sets. And so with that, I will end, and I'm happy to take any questions. Thanks.